It's what you've all been waiting for. You've learned how proteins are made by eukaryotes. Now it's time to compare protein synthesis in prokaryotes, bacteria kind of things, and eukaryotes, in other words, everything else. It's me again, Moser. And after this video, you should be able to compare the process of protein synthesis in eukaryotes to the process of protein synthesis in prokaryotes. And you should be able to explain why it even matters that there are differences in that process. You should already know, the process of protein synthesis in eukaryotes, and you should already be familiar with the basic cell structures of prokaryotes and eukaryotes. Let's go. So you know that prokaryotes, that would be bacteria and, bacteria-like organisms and things like this. And eukaryotes, which would be animals, plants, and fungi, as well as a few complex unicellular organisms, they're called protists, are different. So prokaryotes and eukaryotes, they're different. We knew that, Moser. Well, that's good, because if our cells were just like those of bacteria, it would be really difficult to create drugs that would kill the bacteria, which causes infections, and not harm our cells, or the cells of any other eukaryote, for that matter. What you may not know is that many antibiotics actually target the process of protein synthesis in bacteria. Well, once protein synthesis is stopped, bacteria can't construct the special protein capsules. These are things that protect them from their environment. They also have a hard time building their cell walls, which, while they're not made of protein, are made of peptides, little peptide chains. So no protein synthesis? No bacteria? Yeah, that's about the size of it. So let's start with a very, very quick review of the overall process of building proteins, or protein synthesis, in eukaryotes, like and fungus and earthworms and mushrooms and blue whales and ferns, all those. Overview of the process of protein synthesis in eukaryotes. Awesome. All right, so a eukaryote cell is just, hanging out, minding its business, when suddenly a series of chemical signals, boom, 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 like seriously, 10 chemical signals, causes the process to start. And then DNA unzips, and the information from a gene, which codes for one protein, is copied to mRNA. The mRNA leaves the nucleus, zipping out one of those nuclear pores, and travels to the ribosome. The mRNA gets fed through the ribosome, little amino acids are transported to the ribosome and linked together with peptide bonds. And when the ribosome reads a stop codon, that would be that thing, the amino acid chain is released. It's a polypeptide chain at this point, and it can go get folded and become a protein. So as you probably remember, prokaryotes, like bacteria, have cells that don't have a nucleus. So do they even have to use mRNA? I mean, there's a ribosome, there's some DNA. What's to stop it from just scooting over to the ribosome? Well, let's think about that. There are some really important differences in how bacteria build proteins. Bacteria do, in fact, use mRNA, but since the mRNA doesn't have to leave the nucleus or exit the nucleus, proteins can start being built immediately as soon as the mRNA is completed. Another difference is that the mRNA strand in a bacteria carries the code for several proteins at a time. So, when do we make mRNA? Well, when the right chemical signals are received. So in prokaryotes, it's just one or two chemical signals, and then DNA unzips, mRNA is made, and protein synthesis can get going. Now, bacterial ribosomes are smaller than those in eukaryotes, but the coolest thing about them is they actually come apart. It's like two halves of an plastic Easter egg. They come apart into two pieces, and in prokaryotes, a small piece of the ribosome actually travels over to the mRNA. It attaches to a start codon. Well, then the other part of the ribosome clamps onto the mRNA, connecting to the first piece. A start codon begins the process of building an amino acid chain, and then, well, amino acids are brought over and joined with peptide bonds until what happens? Oh yeah, until the ribosome reaches a stop codon, and then the amino acid chain is released. So, as you can see, there are many ways in which protein synthesis is actually pretty much the same in prokaryotes and eukaryotes. In both types of cells, mRNA is used to get the code from the DNA to the ribosome. Both make use of start and stop codons, and in both cases, amino acids are linked with peptide bonds to form proteins. Oh yeah, and both of them use a ribosome. The differences in protein synthesis in bacterial cells and your cells, however, are what make it possible for some antibiotics to attack the bacteria without damaging your cells. Well, that's pretty cool, huh? Okay. Do you feel like you can compare the process of protein synthesis in eukaryotes to that in prokaryotes? Do you know why it matters so much that these processes are a little bit different? Good. Go on to your recap, review, and reflect.